Okay, we will wait 2 to 3 minutes for sufficient participant to join us, and then we will start.
Okay.
Assalamu alaikum and a very good evening to everyone. On behalf of VitaNova International Alliance for Sciences, I warmly welcome all of our respected speaker, distinguished guests, participants for our class number 5 of the Advancement in Molecular Biology course 2026. We are delighted to have you all with us as we continue this inspiring journey of exploring cutting edge advancement in molecular biology. Today's session is on next generation sequencing technologies, one of the most transformative innovations in modern science that has revolutionized genomics, precision medicine, diagnostics, and biotechnology research. It is my great pleasure to introduce our estimated speaker, Dr. Sabina Jafarzada, head of the biology lab at Qarabakh University, Azerbaijan. We are truly honored to have her share her expertise and valuable insights with our international audience. Before we begin, I would like to remind all the participants that attendance and active participation are essential for the international certification. Now, without further delay, I would like to invite our respected instructor, Dr. Savina Zafarzala, to begin today's session. Miss, the virtual floor is yours. Kindly they proceed with the presentation.
Yeah, thank you very much. And I would like to give some brief information about myself. Just to mention, I'm Dr. Sabina from Qarabakh University. And as now, maybe you have no information, the Qarabakh University is one of the newest universities in Azerbaijan Republic. I'm the head of biology laboratory, PhD in biology, and my postdoc academic discipline is microbiology. It is not some direct related, but it is some relation about my postdoc research. Okay, my today's topic is called next generation sequencing. First of all, I would like to say we have some information. What does mean this process? And now today, molecular biology, diagnostic, the some medicine processes are very important. This process that we can use this today. The introduction, the how it works, the major technologies, and application challenge the future. And and firstly, I would say that the next generation sequence, what does it mean? And this refers that group of the high throughout DNA and RNA sequencing technologies that allow millions of nucleic fragments that we can say that sequenced simultaneously. And there is some increase the speed or the some that reduce the cost of genomic analysis. And it is the first slide that briefly that sequencing Audacity is the robust platform that has enabled the sequencing thousands of millions DNA molecules. It is the some of the high throughout sequencing that has of time used number of different modern sequencing technologies. How we can say that principle that involves there are the DNA and RNA extraction, fragmentation of nucleic acids, library preparation, and massively, let's say, parallel sequencing that we can extract by informatics analysis of the generated sequence data. And from the figure that show that the first step is DNA extraction, and step 2 is the library preparation, and the next is the sequencing, and the final is the analysis of the data. And beginning this process, we can show that the DNA sequencing, that's the parts one, the first part, it is about their DNA sequencing process. It's just major application. We can say that they are the VGC, with some RNA sequencing and others. And the first part, it is the DNA sequencing, the sample collection. Start with the biological samples, such as blood or tissue, saliva, or some teeth, or the biological material. The second, DNA extraction. The DNA is isolated from the sample. Then DNA fragmentation. It is the sum of the smaller fragments just become broke. And library preparation, amplification, sequencing. Sequencing that machine reads the DNA-based ATCG. And data generation, it is the sum that produce millions of DNA reads. And bioinformatics analysis, it is very important for the sequencing data. It is the computer program process to analyze the sequencing data. And finally, it is about the genetic variants, the mutations, and other biological information that we can identify this process. And how we can say that's mentioned that there are some VGC, VIS, and other process. But firstly, it's very important the Sanger sequencing. The Sanger sequencing is about the method that identifies the order of the nucleotides based in DNA-based chain termination by nucleotide called dideoxynucleotide triphosphate. It is the some specific molecules, just we can use this molecule. And it is the Sanger sequencing that there are some advantages and disadvantages when we compare about NGC. And the history of DNA sequencing, that is some history date that human mitochondrial genome sequenced from the beginning till the human microbiome project completion reached. And we know that the human genome project completed 2003. After this process, we can improve our sequence processes because it is the whole genome, the human genome projected that begin the analyzer, some the PacBio sequencer, and other parts. And this simple explanation, the Sanger sequencing, the beginning from the reagent, then the primary analog and chain extension, then the dinucleotide, that NTP binding and chain termination. And what does mean? What's the function of this molecule? It is the stops chain extension. And then can use the fluorescent lab DNA sample and capillary electrophoresis and fluorescent detection. It is about the detector. Detector is the DNA sample can use the laser in the fluorescent detection. And finally, it is the sequence analyze, the data analyze. Just we can use this reconstruction of nucleotide. And this other explanation that Sanger sequencing and the second generation sequencing. What does it mean? What is the difference between them? That is the amplification of region of interest by PCR. And the other part that sharing the genomic DNA and negation of adapters. And it is about the chain termination and some process of this fluorescent level the MTPC, and we can use the both of them in this process. But both of them can use the fluorescent labeled nucleotides, that is the highly sensitive cameras, and record the sequence of light. Is the characteristic of second generation sequencing, and gene 1, gene 2. And why is NGS better than Sanger? Now today's laboratory, the modern biological laboratory, the molecular biology, Sanger is a good, but there is some comparison the NGS is better than Sanger. What does it mean? Why is this very important, and what is the difference between them? And firstly, that's how it works. Works that simplify that PCR amplification, the how it works from the Sanger sequencing. The second next generation began the DNA fragmentation. And what can I say? The Sanger sequence, it is the first generation, the sequencing method based on chain termination. But next generation NGS, it is the advanced sequencing technology that analyzes millions of DNA fragments in parallel. It is very important statement that we say millions of DNA fragments in parallel. And because it is the high quality, better than Sanger. And this some chain termination, the capillary electrophoresis and sequence read. But next generation have the library preparation. It is very great part in this process. The library preparation, this is massively parallel sequencing and bioinformatics analysis. And this process increases our quality for the sequence technology. The library preparation and bioinformatics analytics, it is the only the NGS. And there is some difference that low throughout but high throughout in next generation. The limited scalability but highly scalable, higher cost per base, the lower cost, the lower sensitivity, the high sensitivity, time consuming but faster result in NGC, and limited data types that are suitable for structural variants and complex regions. But when talking about the NGC process, we can say the data support SMPC, NDL, CNV, RNA-seq, methylation, and more other processes, because we can say that some versatile application, not that some the limited data types. Okay, but there are some molecular diagnostic advantages and disadvantages, NGs, and we were talking about some laboratory working or some experimental. We should know there are some advantages and disadvantages because it is the laboratory work, and we can expect some limitation in this process. And because of say that advantages is a rapid, but disadvantages is costly. Sensitive, but lack of specific science for skill. The excellent for compromised patterns, but misconception. And there are some parts that we can say that there are some disadvantages, but advantages are identification of novel pathogens, identification of multiple pathogen infection at the same time, but there is some storage of huge genome data. Maybe there's some process we can lose this part of gene, and it is some complexity of data. And this slide is about the genetic overview NGC. Just you mentioned that there's some difference there from the first, second, or some Sanger sequencing. And genetic overview of NGC is library preparation, clonal amplification, and sequencing. A library preparation is about the input DNA fragmentation, the repair and adapter ligation fragment. Okay, it is okay. But the clonal amplification is the sum each fragment. We can take the two types of an emulsion PCR or the bridge amplification. And because of we can say the library preparation is about the DNA is fragmented and adapters are added. Clonal amplification, the DNA fragments are copied to create many identical copies. It is very important statement that clonal amplification we can say that create the many identical copies. And sequencing DNA TGs are identified, and signal detection. It is the fluorescent or any the luminescent signal are detected. And that analysis of the DNA sequences, and is the DNA library amplification sequences and data analysis. Okay, let's talk about some types of NGC. There are three types of NGC, just mentioned that's whole genome sequencing, or should name called VGC. VGC is about the sequence the entire genome, that means the whole genome sequenced. There are some whole genome that coverage all genes and non-coding of DNA. Cause highest cost and longest turnout time. And the second type is called with whole exome sequencing. There is some difference in fragments. Maybe they are the exon 1, 2, 3, or the some n exon, but they are not any parts or any non-coding. They are sequenced on the exons protein coding regions. And because of that, there's some limitation. Okay, accuracy is good. But entire exome, the only we can say that entire only the exome. And time the some long turnout time, the cost of the effective and depth of the sample is more cost effective than VGC. And the last is called the targeted gene panel. Targeted gene panel, there are some different gene and focus on a specific group of gene. They're faster or more affordable. It is the high sequencing depth, but there are some limitation that may be most cost effect, but accuracy is high. It is the difference between them. But we can use we can use only some specific group of gene, not the whole gene, not the some exons. There is some difference between. And NGC workflow that we mentioned about this, that sample releases, capture and cloning of DNA and deletion. Firstly, template preparation or DNA, cDNA, library preparation, that DNA adapter relegation, and library amplification and cluster generation. It is the some process of NGC workflow. Because we can say that briefly that sample preparation, library preparation, target enrichment, and input sample QS, intermediate sample, and final library. Okay, it is one of the example about this. This is genetic. Maybe we have some problem, or we can should solve as molecular biologists, and we can make our group, that research group. And what does mean this process? Firstly, does this slide explain that is this disease is genetic? We can analyze the family history, age, and at onset, that disease is progression, this is a specific feature, and which gene is normal, or the some mutation, or the other function. Second, that is VGC the correct first line test? Maybe we know some information, the family history, but common genetic variant that cause the diseases, that single gene test and unable to detect their alternate genetic, we should or not. Because the next we can use the providing adequate and correct information, HPO terms and virtual panels and VGC consent. It is very important that genetic testing right for this patient or not. The patient autonomy, do they want the diagnosis or not? Implications for family members and therapeutic decision. It is the sum including unwanted and incidental finding. And because we can use the NGC about these processes. There are the other types of NGC process, the axial NGC, traditional and random priming. There are some similarity, but nowadays we can use the axial NGC or the random priming. What does it mean? Why we can use this? First of all, I would say that all that can use the bisulfate conversion. Bisulfate is the then adaptase, other processes. Traditional is the DNA fragment and repair and ligation reactions. We can use this some library molecules. But random priming, just we say that from the name, they call the random. Because random primate fragment is not the unfragmented DNA. It is the random primate fragment and inefficiently primate fragment. 3' tagging, and then we can use the functional library molecule. And there is some difference just Bisulfite conversion, it is okay. There's some extension in the axial NGC. A broken library molecules is the traditional, they are not the high quality. A random primer, it is the inefficiently primed fragment because the randomly primed fragment. Because the metal sequence is very important and available for this process. And the Illumina sequencing steps. Illumina sequence step is one of the type of the NGC. Just there is some reputation that same processes, the library preparation, the DNA library bridge amplification, the sequencing, alignment, and data analysis. But there is some short amplification that's fragments, adapters, and attach the follow. And then we can use the binder primer, we can use the some type of the PCR and dissociation, the cluster, and final will be the signal scanning. And steps of ion torrent sequencing. It is the other type of NGC, just we use in our today modern laboratory. That firstly, it is the DNA extraction and library preparation. And what is the difference between them? That we can use, there is some chip well, that bead in the chip well, not the other some the illumination or the some the light source, because it is the ion torrent sequencing. And isolation of genomic DNA, the DNA extraction and library preparation. The second, the amplification. The third, the sequencing, that be the chip well with flooding of DNTP. And next, we can use the assembled sequence, the some the overlapping regions. It is the called the data analysis from the bioinformatic sources. And other part of this process, it is called the helical single molecule sequences. There is some advancement that helical single, that's it is the some repair ends at the some ligates adapters that selected their DNA, then attach the DNA flow cell, it is the cluster generating, and then annual sequences primer. And the sequencing the meal will be the some step about strand that generate a base cell. It is the helical single molecule sequencing that we can use some part of process in this station. And it is the SMRT bell template that SMRT is supposed the Zembo wells, the star sequence takes place. It is the hairpin. And the first SMRT, just can say that they are the two hairpin adapters, all a continuous sequencing. Then it is the where sequencing takes place, the labeled nucleotides. And all the DNA types are labeled and available for incorporation. And then same can say the polymerase, the modified polymerase. It is the polymerase, the camera records the emitted light. And then PacBio. PacBio is about the next step, just say the camera records the changing colors from the SMV. Each color change corresponds to one base. And what is PacBio sequencing? It begins from the SMRT bell template and the primer, the DNA polymerase, are going to process the DNA sequence and the hairpin adapter primer, and it is the PacBio system. And it is the one of the species disease system. And Oxford nanopore sequencing. Oxford nanopore sequence, they are the difference between the PacBio, but it is the some minion, the gridon, that flungal, promedion, the nanopore sequence, just we use it in our laboratory. And Oxford nanopore sequencing steps that begins from the sample preparation. It is example nasopharyngeal swab. We took the nasopharyngeal swab, their RNA extraction, then the cDNA synthesis. The second part is we can use the PCR, multiplex PCR, and barcoding. That big barcoding, this process, genome, the primers, the amplified. Then we can go library preparation, the rapid barcoding kit, and it is the some we can use the normal kit. And then sequencing process, that sample pooling and library loading, it is the minion sequencer, that little part of the sequencer. And there is some active pores, the nanopore we can use, the nanopore, the base calling, then the nucleotide, the assemble and analysis. And after this, the processes, it is called the sequences genome and phylogenomics. Why the phylogenomics? Because we know the phylogenetic and dynamic process of genes, nucleotide. Short read sequencing and long read sequencing. And there is some reference genome. The short reads it is about assembled genome. They are not the whole part of the read sequencing. And they are assembly of the overlapping DNA sequencing. And we can use MySeq sequencing, the library preparation. And next, the assembly sequences that showed, that maybe there's some repeated processes or the but show tandem. And genomic DNA from a mutant library, it is the some comparison processes short and long. Short is the sequence, the alignment around the repeat elements, but long read it is the repeat elements, but long of the parts of the library preparation, because they can say they are the same, but they are different the quality or the quantity. Short reads and long reads. Maybe we can take any diseases gene, the reference genome, there is some sequence gaps, and there is some difference that missing sequence that leads to gap in genome coverage and limits variant detection. There is some limits variant detection. But when talking about a long-read reference genome, there is some uniquely and span-large variants providing comprehensive variant detection. There is some difference, the comprehensive but limited variant detection in short reads. And VGC and VC in this process is can say that's whole genome or the some parts of genome. Whole genome fragments that reads that all gene and non-coding DNA, just you mentioned about this. But this is only the exon that coverage entire exon. The accuracy is good, but VGC accuracy is low. The versus is the sequence nucleotides within the entire DNA sequence, but with it is only the exome that is protein coding region. It is not as mean about that whole genome. And full spectrum, but select spectrum, the coding DNA region, just to mention. And when inside the RNA discoveries are made, new insights require resequencing. Sequence once analyzed indefinitely, but there are some the protein coding insights. Okay, and next slide is about some technologies in human and microbiome. Just we mentioned that genomics, the VGC, WIS, transcriptomics, the RNA sequence, and epigenomics, a middle chip and ATIC sequence, and others. And there are some technologies we can use, some parts of data analysis. VGC, WIS, we can use the permutation, the small indels, couple number relation, structural, and range identification. Transcriptomics, just RNA sequence, we can use the gene fusion, differential expression, alternative splicing, and RNA editing. And some of the methylation processes, we can use the metal sequence, histone modifications, the transcription factor band, we can use the chip-on attack. And microorganisms, the microbiome, the metagenomics, we can use their 16S RNA, ITC shotgun tagged panels. That we can use the bacterial profiling, fungal profiling, and 16S RNA, we can use bacterial profiling, but ITC shotgun, we can use the fungal profiling, and the rest can use the some parts of microbiome profiling, emergent profiling, target gene analysis, and others. And all of these technologies, we can use some applications that are functional effects of mutations, network pathway, and integrative analysis. Okay, that is some RNA sequence type, that bulk RNA sequence and a ccRNA sequence and special RNA sequence. And bulk RNA sequence is the all tissue homogenic, that what we can use their tissue level transcripts profiles for the any brain tissue or some other organ tissue. And it is the some barcoded RNA from single cell. We can use the single cell, not the all cell. Only the one cell we can use what and which to determine some cell heterogeneity. And so it is the special RNA sequence is about this tissue section, but determine resolve transcriptional patterns around tissue regions and cellular niches. Niches, it is very important that we can detect tissue regions and cellular niches, but there is some limitation, that's resolution throughout trade-off. And ssRNA sequence, the selective cell nucleus recover bias. And bulk RNA sequence, maybe we can use the all tissue homogeneity, but masking of cell type specific changes, because we cannot identify all cell as specific. Okay, the next slide is about the genomic DNA library preparation, the ongoing processes. They are the subject 200 for 46 subjects, and here's the some stage, the first, second, and third, the quality control. At the last of our experiment, that is very important, the quality control. And because the genomic DNA library preparations, okay, the next, the target DNA enrichments, then the paired DNA sequencing, alignment, human reference, and variant detection depends on the which subject we can use. And the last is the annexation, that's the process, some data analysis, the CAD or the exact and others. Okay, this process is about the some NGS data that's sorting or some the PCR duplicates data removal, best quality calibration. We can use the quality, some adapter trimming, alignment, mapping, other the process. An application of NGC. Summarize of our slides can say the how can application that can use this process. It is the NGC application, the genome sequencing, forensic genetic. In our today's very important forensic genetics, clinical applications, single cell sequencing, and agricultural, environmental applications, and metagenomics. That's very important in our today's metagenome analysis. It is not so easy in our today's laboratory. And transcriptome analysis and epigenetic profiling. Okay, and cancer research, how we can describe the NGC. It is one example, for instance, breast cancer. Breast cancer can use the diagnosis, some the surgery relapse and some 5 years the adjuvant therapy. That tumor tissue collection, that surgery section, blood collection, that detection, the some dissection processes. We can sort the monitoring this process from the blood sample, DNA extraction. It's very ongoing research, but there is some successful news that breast cancer or the type of the organ or tissue cancer, we can identify NGC and prevent some diseases, maybe the genetic or the disorders that they are not related to the genetic diseases. And this is this slide, the cancer genomics stage is a whole genome sequencing that we shows that a whole genome sequence, the VGC, the variant calling, they're using Linux. Linux is the one the program in the as we can describe as Microsoft program that we use this recruitment the sample collection, then exam the capture sequence, the result identification of variants, the some the computer we can complete this program on in Linux, and then we can use the final variants when we're talking about some explanation cancer genomics, some explanation how we recruitment this collection because Linux is the one of the program. And NGC, it is the briefly the explanation that NGC, how we can describe and when we can use this process. That transcriptome analysis, germline mutations, somatic mutations, chromatin epigenetic modifications, and single cell sequencing, and synthesis and 3D genome organization. There is some briefly information about the parting the mRNA, small ncRNA, that is the study of mutational process in tumors. Because the biomarkers or the some gene expression, this information is very useful. And clinical oncology, next generation sequencing is okay. They are the good for our results, but there are some disadvantages for clinical oncology. And first of all, we can start from the advantages, the comprehensive genomic profiling that detects multiple genetic alterations. It's okay. That's view of tumor biology. And personalized medicine is the treatment plans by identifying actionable mutation that's patient response to targeted therapies. Early detection is very important for save life. Identifies cancer-specific mutation even in low levels, aiding the low diagnostic and monitoring. Cost effective is the reduce the cost compared to the sequence single gene testing, as multiple mutations are screened in single round. And treatment monitoring is the facilitated tracking of tumor evolution and development of resistance. And high sensitivity detects the variants and mutations that might be missed by traditional methods. And disadvantages. Disadvantages is data interpretation. Maybe there's some the complex bioinformatics tools to express and analyze this most sequencing data. And the variance uncertain significance that difficult to determine clinical relevance of some genetic alterations or some technical limitations. NGC may struggle to detect structural variants like large insert and deletion, gene fusion, and turnout time. Processing and analyzing NGC data can be time consuming. And maybe quality of sample and biopsy sample or some compromise result may not be so high quality. Or there's some legal or ethical concern, the issue problems, the privacy and data sharing, some incidental findings, maybe they cannot explain our the idea that would complicate the clinical use. And some cost and accessibility that multigen testing NGS is still expensive and may not be accessible to all clinical settings. And it is the molecular diagnostic of tumor that there are some challenges, not the disadvantages, not the disadvantage. We can say the challenge because it is the difficulties, not the problems, not the some disadvantages. Advantages is the massively parallel sequencing capability. We mentioned it. As single input DNA, RNA, and simultaneous screening of multiple genes, it is okay. And better picture of tumor heterogeneity and quantitative and sensitive detection, decreases sequencing cost per gene, and constantly improving technology. And what are the challenges? It is the high complexity, just to say that some maybe bioinformatics or the some deficiency workflow the results. And it is about the validation of assay performance. And maybe some selection genes for the NGC panel, selection some approach sequencing platform, an informatic challenge for analysis and clinical reporting, revalidation of upgrades, and long-term storage or retrieval of data. It is last but is very important, the long-term storage, because it it is a challenge, but we can use some disadvantages or deficiencies of this process. Okay, this is about that one example that tumor tissue or the some blood liquid or the FFP tissue, the body fluid that we can use this NGC process. Some just to mention applications, the genomics, epigenomics, transcriptomics, and proteomics, and metabolomics. That this process ongoing research and some integrated AI. We know that we cannot describe our today modern life without AI. And using AI and the ML platforms, we can make some predict or some process of the mutation analysis, some biomarker prediction, some network pathways, or some research diagnostic therapeutics, just helping from using the AI and some ML platforms. And last, the final slide is about this one simple sentence. Just NGC is not just a technology for reading DNA. It is becoming a technology for predicting, preventing, and personalizing healthcare. And once faster plus cheaper sequencing, genomics becomes accessible to more people. Precision medicine treatment can be selected according to individual's genetic profile. And AI plus NGC, massive genomic dataset, can be interpreted faster and more accurately. Single cell or long run sequencing plus a deeper understanding on diseases and biological systems. And real time portable sequencing, it is the some rapid diagnosis and outbreak monitoring anywhere. Because we can say the final that the future of NGS is not simply about sequencing more DNA. It is about turning genomic information into better decisions. Okay. I finished my today's slide presentation. Do you have any question? I can hear you. It is some information that not an experiment, my personal experiment, but by the way, I can say, and I have my work about the microbiology, some bacteria that is the Qarabakh. It is the extremophile organisms. Maybe next time we can use NGC, identify these processes, identify or the some long-term or short-term reading, single cell, some sequencing, and some process that we need some cheaper sequencing processes for genomics. And because of we sent our products to the United States of America, Niagara University, and there are some process ongoing research that long risk sequencing and metagenomic analysis of extremophile organism, which I mentioned is the Su, Kalbajar, one of the part of Qarabakh. It is the thermal water, and maybe we can say that there are some biotechnology applications. We should know there's some ecological characteristic and biotechnology application in this process, because we ongoing research and we are waiting our expected analysis of what does mean the massive genetic analytics or the some the treatment can be or other the processes. Thank you very much for your attention.
Thank you so much, Miss Sabina Jafarzada, for your amazing presentation and great explanation. Our participants, if you have any question or any kind of confusion, kindly write in the comment box so that we can take that question to our respected speaker so that they can answer. Kindly, if you have any kind of questions, write in the comment. Okay, if you have any kind of question related to the session, kindly write in the comment box. Okay, we have a question which is what AI tools complement the NGS? Would you like to answer it, Miss? Miss Sabina? Yep. Take the floor kindly.
Okay, sorry. Okay, what AI tools complement the NGC? I mentioned that AI tools, we cannot describe our life, that AI, in our education life, the some research life. And when we're talking about the some sample DNA extraction library preparation, this is a sequence data, but when we can use the AI, can help analyze or classify or predict and interpret that data, and we cannot solve about the, any time or the other thing, because the AI it can predict or the interpret or classify, because in this academic presentation we can use the some AI tools for NGC with their purpose, advantages, some limitation, and some process that is very alignment process and AI ML analysis, that variant interpretation, and it can help to us the biological or some the clinical conclusion that predict or the some the alignment process.
Okay, we have another question which is what is BAM file?
Okay, it is the BM file. It is the index that it is the created alongside the BM file. A lot software such can use IGV or the some quickly access reads from some genomic region without reading the entire BM file. It is the some reads mapped to a genome. Maybe FSTQ or other sequence reads. It is the some reads mapped to a genome, the BAM, it's called short variant.
Okay, we think we do not have any question left. I would like to thank you, Dr. Sabina Jafarzada, for delivering such an insightful and inspiring session on next generation sequencing technologies. Your valuable knowledge and practical insights have helped us better understand the impact of NGS in modern technology and molecular biology, as well as genomics. We also extend our sincere gratitude to our participants from different countries of your active participation and enthusiasm throughout today's session helped us to teach more enthusiastically. As a gentle reminder, please complete your attendance requirement, which will be shared in the group. How are you gonna do it? So follow the instructions and fill your mark your attendance that way. And we look forward to welcoming you to our new next session in the advancements in molecular biology course to 2026. Until then, keep learning, keep exploring, and keep advancing through sciences. Thank you, everyone, for joining us. Have a wonderful evening. Miss, would you like to give a closing remark as well? Miss, would you like to take the floor and say something about our participants?
Just you mentioned me about this.
Okay. So, as we are in the end of our session, would you like to say something for our participants who joined your session?
Okay. Lastly, I would say that to our this end of the session participants, that thank you for your valuable participation, time, and your attention. And we truly appreciate your contribution to the session, and I would like to say it's very important for me that this is ongoing research because I am the onset in NGS basic molecular processes, and your the sound question made this session meaningful, and we hope you found informative and useful. Thank you, and we look forward to seeing our next some meetings and some discussing processes. And thank you very much for all.
Thank you so much, Miss, for your meaningful speech. And thank you once again to everyone who joined us today for the session. Allah Hafiz and have a wonderful evening.
Okay. Thank you very much. Bye bye.